I am very, very pleased now to introduce Professor Jay Gild. Professor Gild is a child psychiatrist. He's seeing patients, but he is also an excellent researcher, and he belongs to the National Institute of Mental Health. And they have, I can say that the most experienced research into brain development of average children and of children with disorders of different kinds. So you will now hear an overview that you cannot hear outside this room, at least today. Please.
A great honor for me to be here, being on the same forum as Professor Rudder. It reminds me of the first time this happened. My wife is also a child psychiatrist and saw the lineup and our names there, and I could tell she was very impressed, so I triumphantly exclaimed, Gwen, in your wildest dreams, did you ever imagine that I would be asked to give a talk along Sir Michael Rudder? And there was a awkwardly long silence before she answered, actually, I've never really been in my wildest dreams. So with that introduction, I'll begin talking about sex differences, male-female differences in development, which is not a new issue. It's received a lot of press lately. Larry Summers, the president of Harvard University, got into some hot water about some comments, and it certainly seems to be kind of goes in cycles of importance, but a glance through the literature shows that it's really a quite enduring issue. The very earliest writings, the Sumerian cuneiform tablets, speak of male-female differences. All major religions, prominent in advertisements in the U.S. This is a yogurt commercial, female-centered, and the Manwich, a very fat-laden burger that Burger King is promoting. One of the longest-running comedy theaters in the U.S. focuses on the behavioral differences between men and women. Seem to be about a book a month in educational literature or popular literature focusing on male-female differences. Even board games now, and the men from Mars, women from Venus. So it's a very prominent theme. But the focus on these differences between boys and girls, men and women, has led to discrimination, oppression, suffering, lost educational opportunities, not in our distant past as we speak. Horrible, bad things. And so this raises sort of non-rhetorical question, why mess with this? Why risk presenting research that may be misconstrued, misinterpreted, misused to even make this problem further? And so it is something that we've thought a lot about. What if you do good science, but the end result is bad for society? I think as a child psychiatrist, though, we can't ignore it. Everything that my guild looks at is different between boys and girls in terms of age of onset, symptomatology, prevalence. And these differences in healthy boys and girls might be part of the knowledge about why these illnesses are so different between the sexes. And genetic studies further this notion out. So many of the genes are differentially expressed between males and females, not just in mice, but humans as well. So I think we ignore these differences at our peril. So we need to, I think, do the science as well as we can and try to avoid the overall generalizations. Well, why do we even think there would be a difference between males and females? And I think for me, the most compelling reason is that the evolutionary forces have been different. And I wanted to sort of start with the basics in terms of defining what we're talking about. So sexual reproduction is mixing of the genes. And it's as opposed to asexual reproduction or cloning. When did sex occur? About 1 billion years ago. Looks to have evolved independently many times. About 300 million years ago, after the mammals branched off from the reptiles, a regular pair of autosomes begin evolving into what are now the X and Y. Birds use W and Z. There are different sort of strategies. Where sex, at first in the ocean, now land, sea, cable TV, and the internet. Who, not bacteria, almost no protozoa, but most vertebrates use this mixing strategy. Why? Because it works. It's an evolutionary advantage in the environments for a lot of species to increase the variability by mixing up the DNA. It's not universal. There are these species of whiptail lizards in New Mexico that don't. Many reptiles, they can change the sex depending on temperature or ratio of men to women. And how does sex occur? So the basic strategy here is to get the DNA together. And David Dusenberry, as a mathematician at Georgia Tech, has done some modeling of what would be the best way to get DNA together. And what he ends up with is that the best strategy is for one to stay put and the other to move around. So you might imagine two people lost in a large forest, and if they were going to have a strategy ahead of time, say, look, we might get lost here. What should we do to make sure that we find each other? And it turns out that the best strategy is for one person, this is actually taught in the Boy Scouts and some of these exploring groups, one to stay put and the other to do a systematic search. The one that stays put, the more noise they can make, or in the biological kingdom, sending out pheromones, being big and having all the nutrients, the bigger the better. And for the other, the faster the better. And so these forces then tend to drive to the extremes, where the searcher, the more searchers, the smaller they are, the more effective it is. And what we see then is that one of the variants will end up with very small DNA characters, and that's what we call the male. And the other one will have larger, and that's what we call the female. In a sense, this is what male and femaleness is, by definition, across all the species. And the extremes can be quite great. So the largest single cell in the known universe is now the ostrich egg, but not too long ago, the egg of the elephant bird, which I thought was interesting, described by H.G. Wells from the Time Machine and Invisible Man fame, who was trained as an anatomist under T.X. Huxley. So this was before that work. But you can see the magnitude of difference from a sperm cell to a 2-gallon egg is quite a range of diversity. So for the male sperm, the more intense the competition, the more males vying for the females, the more the sperm. And you can actually predict the size of the testicles across species by the average number of partners the females mate with. So we have these very different strategies. For the male to pass on his DNA is a very different strategy than for the female to pass on her DNA. And things kind of boil down to this basic notion. For females, it's quality, and for males, quantity. And the social implications, I guess, we can deduce and stuff, but fundamentally, that's what the strategies come down to. So they're different. There's different goals. And this difference in reproductive strategy affects many things. It certainly affects body shape and size. is probably the single greatest effect within a species. So if we talk about sex, we need to talk about the birds and the bees, and so we see these extreme examples in a peacock and a peahen, where the male has these dangerously, large tails with lots of these eyes, making them easy prey for predators that really weighs them down. In a sense, they're showing they can squander a lot of resources and still make it. And some interesting experiments, it's literally the number of eyes. So they go in with a photocopier and they actually paste eyes, extra eyes on the peacocks, and they become very much sexier to the peahens, so you can manipulate. And it's not the beauty of them, it's the actual number of eyes that makes them more attractive. And also, we sometimes get some ambivalence, so these hermaphroditic zebra finches who are half male and half female, which isn't so shocking, but the fact that it's left as male, right, this kind of half right down the middle, I think is a striking example in terms of the symmetry and biology. So these are, if you look at it from the left side, it's a male, from the right, it's a female. The brain differences follow as well. In bees, I think most people know by now these huge differences between the queen bee and the worker bees and the drones. This is from my home state. These are both adult, full-grown terrapin turtles, hugely different. And in the extremes, we have this osidex. They live on whale carcasses, and the males actually live inside of the females, only maturing long enough to release sperm. So nature has endless forms, most beautiful, and to quote Darwin in terms of coming up with these strategies. It also affects basic physiology in terms of the five senses are all different in humans at birth. And I stress the at birth part because this is before the social influences and stuff take place, different color frequencies, auditory range, sensitivity to touch, smell and taste are all from out of the womb different between males and females. Behavior, we're getting a little bit more complicated now. So the males in general, because of these reproductive strategies, they tend to compete with each other, display elaborate traits and mate guards. The females will select the males and produce the eggs. And in general, the sex that controls the pattern of mating, which in birds is usually the females, gets the larger brain. And that to me wasn't so obvious. You could think of it would be the competitors that have to be more clever to win the female, but it's actually the female discriminating amongst the males that seems to drive the brain change. So if you only know the ratio of females to males and no other biological fact, it predicts an amazing number of the sexual dimorphism. So just basically how many choices the female has across a large number of species determines a lot of sexual dimorphism or differences between males and females. Recognized from the start, in some senses, by Darwin, noted that sexual selection was different than natural selection, because the sexual selection would be within a species and natural selection among species. But both of these forces are certainly operating for brain size. So there's a sexual selection, but also a natural selection for, again, birds seem to be where a lot of the data for this kind of stuff comes from. But if you look at their innovation in foraging, and it predicts how well they'll succeed in novel environments, it's related to cognitive flexibility, and that's a very good predictor of the ultimate brain size for both men and women. So I just wanted to note sort of both of these, both natural and sexual selection are forces in play. So humans aren't birds and humans aren't bees, so what does any of this mean for humans? Because these differences that they may not have very much to do with cognitive ability. And so Diane Halpern, when she was president of the APA, did a review of this, and I think her bias, if you will, going in, was to not find these differences. But after looking through the stacks and stacks of literature, she came to this conclusion that the differences are there. And I think that most people who have looked at this objectively and critically, it is hard to escape the data in terms of the differences are not these global men are right brain women. Those are always hopelessly oversimplified, but indeed there are differences that are quite robust. So these are the ones that have been replicated the most often. Differences in life priorities on questionnaires, interest in people versus things, risk-taking, spatial transformation, certain types of mathematical reasoning versus verbal reasoning, and variability. So the spatial transformations, a common task is to, in your mind, to rotate these objects. So for the A circles, the question is, are these the same, but just twisted in space? And so I won't try to put anybody on the spot, but you might want to look at A, B, and C, and two of these are the same object rotated in space, and one of them is a different object. And this is surprisingly robust in terms of across many ages, cultures. It seems to be tweaked by testosterone. Males as a group average do a bit better on this, and A and B are the same, and C is not the same. So we have these differences in body shape and size and physiology and behavior. What about the brain? So if we look under a microscope, then I could probably say this is a male brain, this is a female brain. But the tool I use is MRI, magnetic resonance imaging, which is quite nice for studying children because it doesn't use ionizing radiation. So not only can you scan children, you can scan them repeatedly over time. And I think it's really launched a whole new era of adolescent neuroscience in terms of this ability to do longitudinal studies. This is what the brain looks to you and I if we were to take it out of the skull. And this is how it looks to the MRI machine. So this is simultaneously my most condescending and profound slide of the morning, because these gray and white boxes are about 1 milliliter in size. You can get a bit smaller with more powerful magnets and more time, or a bit bigger, but about 1 milliliter. And the white is related to the myelin, shown here. They kind of like little hot dogs, this kind of insulation wrapped around the axon. And the gray is everything else. The gray is the dendrites and the axons where the synapses are, the cell bodies, and the like. So inside of each of these boxes, the best that MRI can do, that we still have millions of neurons, trillions of synaptic connections. And so it's very easy, I think, to over-interpret these pictures. There's actually several papers now sort of on the irrational persuasiveness of brain images. You put them in a paper, even if it has nothing to do with the content, people rate the paper as better study design and more believable. But there's huge limitations to what can be thought of. The airplane analogy is you're coming into Stockholm, you see the roads, and that must be a shopping center, this must be industry. You can tell global patterns, but you can't tell about what people are talking about at the dinner table, which is kind of what you'd want for the neurons interacting. So it has limitations. I think that it's clear now to get beyond these peer deeper into these boxes, we're going to have to go to non-human studies. We've been looking at macaque monkeys in terms of scanning them very frequently. You can sedate them and keep them in the scanner for longer periods, scan every month of their life. So we're looking at one male, one female. I wish it was more, scanning them each month. Also looking at the effects of the group, good and bad parenting, and with or without Prozac. And so lots of children, US for sure, I'm not sure about worldwide, receiving medicines like fluoxetine, serotonin medicines, very little data on the effects of stimulants or antidepressants or other medicines on the brain. But this is in the very early stages, so this is, I think, the next decade we'll see a lot of work trying to look deeper inside of the boxes. The study design for my work is sort of non-creative in the sense of we try to get people about age 3 and above to come to our lab, and when they come we do different types of brain imaging, collect DNA on people who agree to it, and do neuropsychological testing, kind of a changing battery. We've over 6,000 scans from 2,000 children. Half of them are healthy, happy, typically developing people, and half of those are twins. And the other half are from different clinical populations, quite a few from ADHD, over 400, autism, over 300, childhood schizophrenia, over 100. But the basic idea is to see how the brain grows in health and illness. And the long-term strategy is to map the trajectories, and just that first step dominated the first 13 years of my career in terms of just sort of what is the path of change for the different brain anatomy and physiology. About 7 years ago, the emphasis switched to, now that we have a rough idea of the path, what matters, what nudges the path for good or ill? Does anything we do affect that? And the guiding intervention part we haven't delivered very well on that. So we're still hoping to be able to have useful advice for parents, teachers, people themselves. But to be honest, I think it's looking at both genetics and imaging in terms of the real impact it's had on day-to-day life has not happened as fast as many of us would have hoped. So I'm going to just describe the meat and potatoes of development before I focus on the male-female differences. And since the MRI sees things in terms of white and gray, the white matter increases throughout childhood, adolescence, and well into adulthood. So the 5th decade for women, the 4th for men. It's not the same slope throughout. We don't see any period of time where the white matter decreases. And so it's much more kind of a ever onward of increasing throughout. The myelin comes from these octopus-like cells, oligodendroglia, mini branched glue, and they send out these tentacles to wrap the insulation around the axons. This picture is actually not correct because each oligodendroglia will only send a tentacle to one neuron, which has implications for information science. And it's not very subtle. So the myelinated axons go 100 times faster than unmyelinated, so about 200 miles per hour at the most myelinated ones. And sorry, let's go back one. The electrical signal hops from node to node. And so the signal itself is a flow of positive and negatively charged ions across the membrane. Then they have to be reloaded, reset for the signal to send again. And so by being able to only have the ions going across at intermittent parts, it's able to reload 30 times faster. So those two together really make a quite whopping difference. We have 100 times the speed, 30 times the turnaround, so a 3,000-fold increase in bandwidth. It's very human. We have far more myelin than any other species. I think it's a big key to what changes during development. So in the early going, we would look at the different brain parts. The hippocampus does memory, the amygdala's emotion, and we had these sort of very simplistic notions of the different brain parts, quote, doing something. I think we could say the same for genes now as well. We say this gene does, then we fill in some oversimplified behavior or trait. So the different brain parts, I think, are better thought of as like letters of the alphabet. And any reasonably interesting behavior is at least a combination of those letters into a word. And as we go from a baby to child to adolescent to adult, then those words become sentences, the sentences become paragraphs, and so on. It's this integration. So the basic letters are kind of there by about 8 months. The hippocampus hooks up to the prefrontal. That's when people can have memory longer than 8 seconds and has implications for stranger anxiety and such things. But the basic kind of building blocks, if you were, are there quite early. And so this notion of increased connectivity of the different parts of the brain communicating with each other more and more has really become the mantra for the imaging studies. So when we look at the total size of the brain, it's pretty stable. By age 6, it's already 93% of adult size, which is actually kind of disappointing when we started this, because we were thinking as the brain got bigger and we'd get smarter, we could match the size to the performance. So you're young, you can do addition, and then your brain gets bigger, algebra, and bigger still calculus. But so it was kind of disappointing that there was so little change in the size of the brain. But when we look not just at the total size, but at the details, then things are actually quite dynamic. So this is the connection between the left and right halves of the brain, the corpus callosum. It's sort of a representative white matter structure. So the volume, or the area of the corpus callosum reflects well the total white matter volume. But we see, this is actually my daughter here from 6 to 7, but in a 1-year period, an 80% increase in the area for that part of the corpus callosum. So globally, yeah, it's like watching paint dry, or very glacial speeds, but at local levels, things can be quite dynamic. So this has really motivated a lot of changes in the technology of MRI to look at this connectivity and look at white matter. One of them is diffusion tensor imaging that people may have seen in the literature. Another white matter one is magnetization transfer imaging, but it's really sort of become the fad or rage to really try to get at connectivity. And one of the outcomes of the increased research on this is that it's not just maximizing speed. It's not just a matter of the faster that the signals go, the greater the intelligence. It turns out that it's all about the timing. And again, this isn't a side dish. This is how information is encoded in the brain. It's how the neurons are firing together. It's the patterns of timing that conveys information in the brain. So this is huge. And the white matter is a part of that. And so, for instance, if the frontal lobe needs to integrate information from all the different senses, it'll get visual information from the back of the brain, and it'll take auditory information more from the middle. If they're going to arrive at the same time, they can't be going at the same speed. And so this has been worked out more in non-human models, this sort of exquisite timing, because if a neuron is going to take in all the pluses, all the minuses, all the inhibitory, all the excitatory, it has to add them all up within about a 50 millisecond window. That's exquisite timing in terms of that to really make the decision. And so this could be a part of the story for dyslexia, ADHD, learning disabilities. It also turns out that as you get more myelin, it inhibits new axonal growth. So you get this almost inevitable trade-off as you get faster connections, but then you lose some of the potential to have more and more connections, fewer but faster. It raises ideas of sensitive periods in terms of, are there certain times in development when we're more able to learn certain things, or visual system is an example of that. If we have cataracts, a clouding of the lens of the eye, if that's not corrected by about 18 months, it's too late. The brain needed that input earlier. And this idea of integration, of going from letters to words to sentences, would rely on the white matter connections. This is during the brain surgery, doctor, what's the matter? Gray from the looks of it. And the matter with gray matter is it doesn't grow like white matter. Not at all obvious. Right? From the moment of conception through death, they're constantly interacting. They're part of the same neural circuitry. Even though MRI sees them differently, that's sort of not really a functional distinction. And so it's a bit surprising that as white matter is ever onward, it's increasing throughout. Not that different by big region like frontal lobe and temporal, whereas gray matter, it's like an upside-down U. Increases during childhood, hits a peak, and then decreases during adulthood, and it's very regionally specific. So if we think of the top of the U as an indication of maturation, not as a peak in ability, I don't think, but just in terms of as a guide, then we can see the different parts of the brain grow at different rates. And the frontal lobes, for instance, then hit their peak thickness at about 11 in girls and about 14 in boys. And in my world, that's a big difference. That's the 3-year difference. And when we first thought, we kind of were running to the educators and they're like, guess what? The girls' brains are maturing faster than the boys' brains. And they're like, duh, we've only known that for 100 years, thanks for nothing. You can go back to your lab now. But for us, it was meaningful that we saw this pretty big difference. And this upside-down U isn't for all the parts of the brain, it's only for the 6-layered cortex, so the most advanced in evolutionary terms. So the 3-layer cortex and that we share with reptiles and other species, and then the intermediate one. So the most primitive, the 3-cell layers are no change through development or flat. The intermediate ones are linear changes, and the 6-layered ones are cubic trajectory. So we see this sort of link between the more old or primitive evolutionary, that's not the right way to think of it, I know, but the simpler the developmental trajectory. So this kind of loose link between evolutionary changes and developmental changes. So what's happening? Why is the gray matter getting thicker or thinner? Are we getting more neurons? Probably not. So in the hippocampus, olfactory bulbs, and cerebellum, yes, and that's incredibly interesting, but it's not driving these changes. It's just the numbers don't add up. So the idea is that it's due to arborization, arbor the Latin word for tree. They're growing extra branches, twigs, and roots. They get bushier, same number but more connections. And then there's this pruning process, which is nature's way of, I guess, something called neural Darwinism. You overproduce, and then you have a fierce competition, and it's a way to ensure that the winners are healthy and robust and strong or useful. This 1-2 punch of overproduction and competitive or selective elimination seems to drive complexity not just in the human brain but in nature. Almost every complex system that emerges can be viewed as this overproduction, intense competition paradigm. And it's not just MRI images of the brain, so post-mortem studies in animals and humans, EEG measures of electrical activity also follow this childhood buildup, decrease in adolescence, PET scans of glucose utilization. This is one of the most cited bodies of work on this issue from Peter Huttenlocher's sort of life work looking at this pattern. And if you look closely, it's conspicuously absent. So the actual data during adolescence, there's only 4 data points in the entire literature. So I don't want to make too much of this, but this is surprisingly unresolved issue, and that the neuroanatomists I've talked to from U.S. and Europe, and nobody really has much data. Adolescents rarely die and get post-mortem work. So this is one of the unanswered questions about what's driving these images. It's even for the receptors. This actually shows dopamine receptors, but also for norepinephrine, serotonin, acetylcholine, BDNF. So lots of the transmitter systems that have been looked at have a childhood buildup, decrease during adolescence, leveling off for adulthood. So I think it's a general phenomenon.
A flurry of growth. Then unused branches or pathways are pruned. And it is this pruning that gives the tree its shape for the future.
So again, kind of condescending, but I wanted to show these because the Zitz cartoon strip is quite popular in the US, and the authors of this have been very generous in sharing these movies and other pictures for people to give talks to schools, and in terms of to try to get this message through that this is a good time, in their life, that this is a time when their actions can have a lot of influence on how their brain optimizes, how it changes, for their adult years. And I think it's, been helpful and stuff because it's hard sometimes to convey science to the masses. So I've been very appreciative of what they've done, and I think that it is empowering for children and teenagers to know that what they do matters in terms of how their brain can grow and develop. So this takes a little while to absorb, but this is looking at the gray matter changes from ages 4 to 22, and the bluing can be thought of as a maturation in the sense it's getting closer to the adult levels. And if we look right here, the dorsolateral prefrontal cortex, it doesn't hit adult leveling out till about age 25. And that was ridiculously late in terms of our, Professor Rutter mentioned earlier about the best of science are the surprises. But this we didn't see coming at all in terms of when we did the study where child and adolescent psychiatrists, should we go to 16 or 18? By 16, they're pretty big. They're getting adult doses of most medicines. It's only since the Industrial Revolution that we kind of made the whole idea of adolescence up anyway. And so we'll play it safe. We'll overshoot. We'll go to 18. 25 wasn't even on our radar screen. It just was way out there. And then we kind of joked. Like when you go to the airport to rent a car, you have to be 25. And we thought, there's the problem right there that the Hertz and Avis, and they have all the best neuroscientists working for them, and no wonder we can't make progress, and kind of joked it away. But when you thought about it, it didn't go away. I mean, they have way more data than imagers have, and they have overwhelming evidence that 24-year-olds cost them a lot more than 54-year-olds. And 24, they're not a novice driver. Right? And you have been driving 8 years, 7, 8 years. Your reflexes are better, your vision's better. If you do a video game, they'll win. And so it was kind of puzzling. And not just for driving, it became the whole kind of paradox of adolescence. You're at the top of your game, tolerance to heat and cold, resistant to cancer. Almost everything you measure physically, you should be in the prime. Morbidity and mortality go up 200, 300%. Number one by far is motor vehicle accidents. So if you're an adolescent girl and you die, 50% of the time, motor vehicle accidents. That's a father of a 17-year-old new driver. It's scary, But the good news is that the rates are quite low. But that's staggering. Number 2 and 3 are suicide or homicide, depending on how you classify the events. All the top 10 seem to be related to judgment, decision-making, emotion. These pictures don't prove that, but it's at least intriguing to wonder about matching this part of the brain involved in these type of things of long-term strategies, impulse control, planning, organization, that rely on this circuitry, this idea of time binding, tying together your memories from the past, the current input, your hopes and dreams for the futures, that this part of the brain, it almost makes sense that it's so late to mature because it takes in the information from everything else. But this is something that certainly we wonder about implications for educators and society. This movie was actually used by the U.S. Supreme Court in 2006 to raise the death penalty from 16 to 18, a 5-4 decision, real close. But what does it mean for the adolescents themselves, for societies, and not that the brain is damaged or broken. It's doing what it's supposed to be doing. I think as an adolescent brain, it's supposed to be plastic, flexible, changing. That's the whole beauty of it, in a sense, is that the human brain has been so adaptable. We can live on the North Pole, we can live on the equator, we can live in outer space with the things that our brains have developed, very changeable. And so at the Smithsonian Museums, they just in May 16th opened a new exhibit on the human origins. And one of their main messages there is that the events driving brain change weren't so much complexity as change. So they look at climate change in relationship to brain growth, that it was survival of the versatile as much as survival of the fittest, that really humans do a very I think that's probably what makes us most human, this very long period of plasticity, and that our brains just 10,000 years ago, we were hunting, gathering berries, if you were in the North Pole, staying, and now we spend a lot of our time computers, reading, doing things that, from an evolutionary standpoint, 10,000 years is a blink of an eye. And we're asking our brains to do such different things. And education-wise, reading is huge, right? And for most people in this room, we spend a lot of our waking hours with reading. Most humans have never read a single word. Okay. Since splitting from the other hominids 6 million years ago, there's been 100 billion humans. Reading's maybe 5,500 years old. So humans that have lived and died, reading isn't sort of natural thing, but our brain is able to adapt and to change. And so I think this is a kind of a key concept for education, that the brain is always plastic, it's always adapting, but particularly during certain times and for adolescents, this part of the brain, in terms of the frontal lobe part, is very active, it's very dynamically changing, and that I think that it will do a great job of adapting to whatever environment we present it. If that environment is violent and uncertain, if it's... the way I see schools is sort of they're creating an environment that the brain will adapt to, that will be good for the next 80, 100 years in terms of what do we want the brain to adapt to prepare best for the adult world, which isn't strictly a neuroscience question. I think that is a much broader question, but I think in terms of the harnessing this plasticity is a key concept for educators and parents. We don't know much about what matters. Is anything we do as a parent matter? What about stimulants and antidepressants and antipsychotic medicines? Diet, video games are hugely popular and different than just 10 years ago. What music, sports, bacteria, viruses, what matters? So that's a big part of what we're trying to sort out, what changes these things. And if we have this overproduction and competition, what if something tilts the battlefield? What if something changes how that works? Could that account for learning disabilities, developmental delays, and completely speculative, but people have wondered about autism, if they have larger brains early in life. What if they have too many competitors for those fewer spots, and that upsets the usual way that the brain optimizes? So a lot of questions from this overproduction, selective elimination, and this was a fortune cookie that I came across that summarizes my entire life's work, which is sort of humbling, but it's this idea that neuroimaging, like life, is about the journey, not just the destination. And an example of that is looking at these images that I just showed. Can we predict IQ? Which is a relatively stable kind of a trait across the lifespan. But these are all healthy kids. The blue group is IQ above 125, so the high IQ group. The lime green is between 110 and 125, above average, and the red group is average. And what to note here is that if we look at age 19, nothing. We can't predict anything from age 19 about that. But if we run the clock backwards, if we go back to see what the path to get there was, then we see the separation. And somewhat surprising, at least to me, actually the blue group, the high IQ group, was the latest to peak. And I don't know how relevant this is to education, but an outcome was that it generated a lot of discussion that I found interesting about these sort of tortoise and hare phenomena. in terms of, is it always better to read at the youngest age, or to be able to do the math at the youngest age? Or do other people, maybe they didn't do it as young, but they keep going, longer? And so that's one of the things I always like to hear from teachers and people in terms of data regarding this, in terms of people who might have been more slow and steady like the tortoise and end up not only catching up, but maybe even going beyond. And what if we categorize children too quickly? Maybe we're missing a lot of people who might be very, quote, gifted in math or the things, if they were given the chance, or if they could learn later. So I think that it raises a lot of questions about sort of timing and pace versus distance for acceleration. When this paper came out, it got some press coverage, but the coverage really missed the point. So we had the West Coast, where smaller brains better, and the East Coast, the larger brains are better. The whole point was that's the wrong question. It's not bigger or smaller. It was the trajectory itself. This was the pinnacle of my career. I made capitalist imperialist pig.com website, and I should have retired then, but I've stuck with it. So this is, I think, it was bittersweet because it invalidated the prior decade of our work, where we were saying you have a big amygdala, you have this kind of straightforward size things that it just is the wrong way to think about it. And we also have these periods then where things are very busy. These times when there's an explosion in the number of arborizations, of different branches, and less so. Very early in life, like for the visual system, when these sensitive periods happen, that's what you see. You see that a lot of growth and then a lot of the competition back down. But does that hold, for later? Does it apply to other things? And so if we look at what is busy like this for the basal ganglia, so this is not on the cortex now, this is more inside, they have this explosion and pruning back between the ages of 7 and 11. So they are involved in a lot of things, but one of them is very fine motor skills, like you would see for music or sports. And if you talk to music teachers or coaches, they almost universally agree. Like if you want a world champion figure skater, you better be doing it before age 11. Even if you were a gifted soccer player before, even if it's not that you can't get really good. You take up golf or tennis when you're 40 and have a wonderful time with it, and all, but to be world class, it really helps to be exposed during these periods. And again, it's not subtle. We see a 50% loss in these BET cells. The basal ganglia they've changed a lot in our recent evolutionary history. And so one of the ways that people look at this, we convert all the species to the same size by convention, a 70-kilogram hedgehog, and now we compare the brain parts. What's most different, not surprisingly, is that neocortex, the outer 6 layers of cells, 156 times larger in us. A huge difference. But in second place are these basal ganglia, and the only thing in double digits. And so, again, as a kind of metaphorically, this idea of moving parts get broken. That the things that we acquired most recently in evolutionary history have less genetic redundancy, less back, and almost every disorder that you can mention, basal ganglia is implicated in developmental disorders. So summarizing, white matter goes up. So we have this idea of increased connectivity in many different forms. So the connectivity can be in terms of the literal connections between different parts, the white matter. It can be connected in terms of, in fMRI studies, things light up together. In genetic studies, that they're connected in terms of they're influenced by the same genetic or environmental factors. Whatever version of connectivity you pick, it goes up during adolescence. And so this could be a key factor of the cognitive and behavioral changes. We have upside down for the gray matter and relatively late maturation of these very high association areas that are tying together information. Very early stages of this notion of critical or sensitive periods. Can we do something useful with that? Can we try to identify things where educators might focus more on different interventions or teaching styles? And the importance of considering the journey, not just the destination. So this concept has really been key because if we look at the journey and the trajectory, it much better predicts cognitive variables, behavior variables, male-female differences, which we'll see in a minute. Heritability changes a lot by age, which I think has big implications for genetic studies. If something is genetic but not even expressed until adolescence, it would affect. It helps predict disease, illnesses, schizophrenia, autism, schizophrenia are all discriminated better by looking at the trajectory than the snapshot in time. So the corpus callosum was one of the first structures reported to be different between males and females, not directly related to reproduction. 160 papers. I did a fairly recent review of this. About a third of them say it's bigger in women. About a third say it's bigger in men. About a third say it's no different. And part of the reason for that, I think, is like in our data, is it different? 4 to 7, yes. 7 to 11, no. 11 to 14, yes again. That the answer changes, and changes a lot depending on the age in question. And the literature will combine corpus callosum size from sometimes from ages 4 to 80. You can completely collapse it across these huge things, which I think is sort of the wrong way to go. So it's nuanced. And if we look at the just size of the brain, one of the blue is boys and the red is girls, about a 10% difference, which isn't accounted for by body size or any other covariates. But here we see these two blue dots. One, the highest and the low, the biggest and the smallest are males. I'll come back to that in a minute. And these boys have 100, double the brain size. Both completely doing well. A huge variance with still good results. So this actually came up during the Pluto planet debates about the importance of size. But when I began my psychiatry training, it was as a Freudian psychoanalytic type, and we used to argue about this question a lot: does the size matter? And then when I switched to the more biological, it was the same question, but we went a bit north to the target. But it is an interesting question in terms of the relationship. It's definitely not bigger is better, as we saw from the IQ data. At the younger ages, actually smaller was better. So I'm trying to remind myself as well to sort of get out of that mindset of just bigger, better. But what is most discriminating is the path. So if you look at the literature, it's incredibly muddled because if you correct for a 10% difference, then a whole bunch of things become proportionately larger in females. If you don't, then a bunch of stuff are larger. And it's sort of like it depends more on what you do about this 10% difference than the data itself. But the trajectories are robustly different in when they reach their peak, especially the cerebellum, which I'm not sure what to make of it. It's the most sexually dimorphic structure we've measured, the latest to reach adult volume, and the least heritable. It has a lot of interesting features for development, and most of the literature doesn't even look at it. You see the picture, they don't even report on anything about the cerebellum. We have structures like the amygdala and the hippocampus, which are very interesting in memory, behavior, emotion. Amygdala is loaded, well, they're both loaded with a lot of hormone receptors, but amygdala mostly male hormone, androgenic, hippocampus mostly estrogenic. And what we see is that in males, the amygdala goes up after puberty, but not in females. In females, the hippocampus increases after puberty, but not males. So it kind of rings true a bit in terms of the relationship between hormones and the brain. So this is looking at the difference between male brain growth and female. I think we had a little trouble switching from Mac to PC, so I'm not sure if the movie will run, but what we see, oh good, I think this one is moving, is that where the differences are, it follows the same pattern where the males catch up in these developmental curves, it's in the same sequence. So the prefrontal cortex area that I showed is the last where the males catch up. So we have this impulse control, long-range planning part where the males are the latest than the females. We have language, social, areas, again, particularly late in the boys. And the parietal area involved in that rotation task that I showed in mental is about the only thing that we see earlier in the boys. So the other thing from the behavioral data that I mentioned was variability, pretty robust across lots of species. Again, that the one with in humans, the XX females versus the XY, the XY is more variable. In birds, it's switched so that the males are the one that have the WW, so they have the same. But it's almost like an averaging effect. But so many things you measure are more variable in males, and what we just saw for total brain size, that holds for almost every measure we have. The biggest and the smallest are boys. And so we can have these differences. These curves have the same mean, but the variance is different, which matters most at the extremes. So if we look at the very tail end of those, we see the difference is the greatest. This is an example for height. If you, I'm not sure whether to use metric or US system, but at 5 feet 10 inches for males, it's 30 to 1. Just 2 inches further at 6 feet, 2,000 to 1. And so this is what got Larry Summers in a bit of trouble, wondering about if the variance is inherently quick. Do you get more extremes? At the very extremes, do you, at both extremes, high and low, would you expect more males? It's robust in our data. So the cortical thickness, the white matter volumes, pretty much everything we've measured. So everything that's not white on this picture is statistically more variable in males. It's in some ways kind of the most robust of our findings. And what accounts for some of this variation, we've just looked at this now. The androgen receptor links, so this is, affects how well your body receives the androgen that's there. You can have different number of repeats. And what we found is that even within males and within females, that it predicts. So the pictures that I just showed and stuff, if you just looked at androgen receptor length, the better and worse androgen alleles, it moves in the anticipated direction. This is kind of a cautionary note on this genetic stuff. We paid 500,000 krona for the egg of a supermodel and the sperm of a Nobel laureate. She didn't turn out quite like we planned. This idea of pleiotropy, that it's hard to pin down sort of if genes are, quote, good or bad. And they may be good at one time in life and different than another, but we're still very interested in it. So if we have these differences, why are they there? So societal influence, they're huge, right? I mean, from the moment the baby comes out, the grandparents, they're going to be treated differently, different expectations, even if you try really hard not to as a parent, the world around you will. So I'm going to not deal with that. Just setting that aside, biologically, we have the X and Y chromosomes changing hormones, and then we have kind of the more direct hormonal effects. So to kind of explore this a bit, we've been looking at clinical populations that have different numbers of X's and Y's and have different hormone levels. So the most common of the extra X and Y's is Klinefelter syndrome. And so this is a non-disjunction during meiosis. They get an extra Y. They have these features that are shown here. But by and large, some of them do quite well. We have doctors, lawyers, computer scientists. They used to be told that they're infertile. We have 5 fathers in our study now through not the usual methods, but through in vitro fertilization, so they can be fathers. So this was kind of worked out pretty much as anticipated in terms of the parts of the brain that were most different in terms of the parts here and here are what we see in ADHD. About almost 80% of the XXY boys are diagnosed with ADHD. We have the speech, language, social areas that look similar to our autism and schizophrenia studies. Also in the expected directions for that. But then we have this parietal sparing. So that was, again, back to the, that mental rotation task that I showed earlier. There it shows empty, but that actually it's not only preserved, it's in a sense enhanced. And they do particularly well in this task. Not only, again, sort of given their IQ and stuff, they do better, they do really well. I'm not endorsing this, but this was like the big news for when they did the autopsy on Einstein's brain. That's where the most different was in this parietal area, again involved in the spatial manipulations. But here we have a group that sort of their brain is better suited for visual learning, for maybe hands-on learning as opposed to reading and writing, and that may have educational implications. But we have all the known variations. So from 1 to 5 x's, from 1 to 2 y's, and each extra x or y you get, you're a bit worse off clinically. But again, that's not super obvious because if you have an extra x, it's inactivated. Most of it. So about 85% of it is inactivated. So it kind of narrows the search a bit because if you are getting worse clinically, then it should be related to that 15% of the X chromosome that's not inactivated. Another pattern that we see, this probably doesn't show up well from the back, but if we start out with the X's and Y's, it's unerringly consistent. Each extra X makes the brain smaller. Each extra Y makes it bigger. So even through the clinical populations. Again, bigger is not better. The Fragile X, autism, the clinical groups, but still, there's a clue there, I think, in terms of how consistent it is in terms of total brain size. The different groups with unusual hormone levels, what we're seeing is specificity. So the hormonal level ones affect amygdala, hippocampus, the more hormonal-laden areas. And so we're looking at all these different populations. Kalman syndrome is interesting. They don't have a sense of smell. And as that's developing, the gonadotropin-releasing hormones ride along the olfactory bulbs on their way, and so if that's missing, so is their testosterone. So they don't have testosterone, but are otherwise healthy and happy. So my conclusion on this is that the male and female brain is overwhelmingly more alike than different. So 6,000 scans, if you showed me one, I couldn't tell you. I couldn't say this is a boy brain, this is a girl brain. So I don't want to make too much of that. What is most different is the trajectories, the change over time in development. The male brains are more variable. That's probably my most defensible statement. But these are group differences. They aren't a constraint for boys or girls. And a metaphor or analogy I can think of, if you say all girls should be in this school and all boys in this school, which is been an argument proposed, if we look at all of the stuff I just showed, all the behavioral data, all the imaging data, and compare it to height differences, the difference between height between men and women is twice the effect size of everything I've just shown. It's pretty whopping, actually. If you take 100 men randomly selected, almost, the average almost every time will be taller than the men. But if you went into a school and said, okay, everybody above 5'6", you go to that locker room. Everybody below 5'6", you go to that locker room. Well, it would be outraged, right? Parents would, well, that's ridiculous. And, well, why? Well, there's too many exceptions to the rule. There's too many girls taller than so many boys. That'd be a horrible way to, But to do the same with just imaging or any single variable would be even worse. So I think that we wouldn't want to just sort of pick one image or something. I think you have to really go back to the individual child. So if you ask me, are the boys and girls' brains different, I hedge my bet. And sort of on group average, yes. What are the educational implications of that? I'm not sure there is at that level. It must be, what is this girl and this boy? And that you could maybe make a decision about what environment would be best. But I think that it's a slippery slope if we sort of focus too much on just that single aspect. And we'll stop there, and hopefully I left time for a question or two, but thank you all very much for your attention.
Thank you indeed. And we will have time for questions and comments. Well, there was an old autopsy when I was in the course of pediatrics. You were always told that a girl fourfolds the brain volume from birth to the age of around 11, and boys not until 14. And that had implications for when you had the growth speed and the start of menarche girls. And so do modern MRI support which you showed that there are different periods when there are differences, but other periods that the differences are maybe not there.
Yeah, it's a very intriguing question, the relationship between physical body growth and the brain growth. When we first saw that, it was easy. 11 in girls, 14, of course, it's puberty, it's like a direct hit. Except when then we looked at the individuals. And so the girls that have later puberty or earlier puberty, it doesn't go along with the cortical thickness. And so we're trying to sort those out now. So certain things, yes, amygdala and hippocampus, decision-making, the striatum, these parts of the brain, they're more hormonal. Other things are more age itself. And it is sort of fun to separate them. So decision-making, the big thing, after puberty, you can predict most of boys' decisions by will this lead to sex. In terms of, even if they don't think of it in those terms, it predicts a lot. For the girls, it's a little bit different. Will this lead to increased social standing? But those are both sort of puberty effects. If you have them do a logic problem, that doesn't seem to be. So whether they've had puberty or not, it's more are they 14 or 12. And so we're trying to find that link in terms of between outside measures of brain change, because if we do try to use the imaging for educational implications, it'd be great to have an easier marker of what the brain is doing. So a long answer to say we don't know. We're pursuing that and wondering what are the links between the growth spurts and the brain spurts. And it's turned out to be sort of a nuanced answer behaviorally and imaging-wise.
And there were also, when following children, you saw that girls had a kind of a development when it comes to social competence and to intellectual competence starting school, around 2 years ahead of boys, but boys were catching up from the age of 15, 16, so there was an evening out of sex differences. And how is that relating to...
10, 11, 12-year-old girls are often interested in boys and interested in reading magazines about cute boys, and there's a definite lag where the boys are nowadays much more interested in the video games and doing things. It's right around 5th, 6th grade, it's striking, in terms of the different sort of, again, lots of exceptions to the rule, but mostly. The catching up part, again, before I used to think, yes, but it is interesting from preschool to kindergarten to graduate school, as a group, the girls do better. So, Kathy, if you had to choose from... so it is interesting in terms of, is there a catch-up period? Because even in late adolescence, there's a huge difference in violence, aggression, incarceration for males. In terms of, in that aspect, they actually get farther apart in late adolescence in terms of some of the behavioral measures. But intellectually, I'm not sure, actually. It'd be interesting to note in terms of, is it a delay or a deviation? If the part of the brain involved in things like delaying gratification, I'll give you $900 now or $1,000 in 3 months. that is, the answer to that changes all the way to almost 30. So people, get better at delaying gratification, way beyond what I would consider, adolescence or anything. So these are, we're really struggling to find out what actually does change, because when we bring we do a Turing test from computer science, where you have, on the other room, you have either a computer or a person. And you can, just by asking a question, you have to figure out which one it is. What would be a Turing test for a teenager? What would you ask them that you couldn't see them and stuff? What would you ask them that by their answer you would know? It's very hard. We've as a thought experiment, we've wondered what would help with that. So in the laboratory, they're very smart. They assess risk very similar to adults. It's just that in their real world, with peer pressure and high emotions, that their decisions are different, and the brain imaging is different. So it's like a different system when you have a peer present for the task. They become more risk-taking, and it depends on if the peer is a boy or a girl or a parent. And so we're just barely beginning to really kind of get a handle on this difference between hot and cold cognition, where the hot cognition is in the real world, and that's what baffles, I guess, parental people. It's like, what were you thinking? And they don't really know, and I think that's probably honest. If you badger them enough, they'll come up with something, and you say, aha, you weren't thinking of the consequence. But it's usually not that. They don't wear a helmet if they're skateboarding. It's not because they think that their head's not going to break, which was kind of what I was taught, that they think that they're invincible, or they really don't. If you really ask them, they don't think that they're superhuman or things like that. They just don't really think about it. They just do it. And so I think that it'll be, the decision making is key, because that's unwanted pregnancy, substance abuse, so many of the things basically boil down to how teens make decisions. And I think, other than just say that we have a long way to go, but I think that's where a lot of the well-placed interest is in, to see if we can learn from the economists and people who are really good at decision-making theory and practice, to apply that to teenagers, because so many things that really matter for teens, certainly, just like all those things do matter too, but ultimately the decision-making is what really can impact their life. Car accidents, unwanted pregnancies, sexually transmitted diseases, these I try to focus more on the irreversible type of decisions.
Thank you. Well, then we have reached the end of today, and now the panel will go to have a kind of a brain work, and all of us, we will go to have a more relaxed time. But someone has lost a wallet. Någon har tappat sin plånbok. And Helena has a wallet. So if you feel that you don't have money for the tube back home, please go to Helena. Well, Jay, thank you indeed. Thank you.